Hello students, this is Dr. Faizan Mirza. In this video, I'm discussing the principles associated with genetic technology. This is what we discuss in chapter 2, chapter 8 of A2. So what are we discussing here? These are the principles of genetic technology, the basic principles that are applied when you are discussing genetic technology. And one of the basic procedure that we do is basically having several techniques that we discuss one after the other. For example, we carry out gel electrophoresis. We have to obtain the gene. What are we going to do with that gene? Where we are taking the gene from? Where we are placing the gene? Where the gene goes, and how actually is going to be useful for the cell in which the gene is being placed in. So let's start. So the first thing is we have to obtain the genes for genetic engineering, and normally we obtain the gene from a cell and we separate it using the process called gel electrophoresis. Gel electrophoresis is a technique for separating the DNA fragments on the basis of size, and we can pick the gene or you can say the fragment of our interest. So once we have separated and identified the DNA fragments, this can be used for forensics, it can be used in genetic technology. What exactly is genetic engineering? In genetic engineering, we deliberately manipulate the genes of a cell or an organism. This can be done in one of the three ways. Either you directly alter the genes of a cell, or you directly remove one of the gene from the cell. So the cell cannot produce that kind of a protein because the gene has been removed, or you transfer a new gene into a cell altogether. So these are the three basic ways in which genetic engineering can be done. So if we take this further, what stages of genetic engineering do we continue with? So when we're discussing the stages of genetic engineering, there are as good as five different stages here. So these are stages of the basic five stages of genetic engineering. These include, first of all, you have to obtain the gene. When we obtain the gene, we have to take the gene out so the desired protein can be made, and we can obtain the gene using gel electrophoresis. It will help you identify the DNA fragments. Then you have once the gene has been obtained, the next thing is that you make an abundant copies of those genes. So you have a lot of DNA sample with you of the same sort, one gene and many copies of those. So this basically is done using a technique called PCR that amplifies the DNA fragments. Once the DNA is being taken out and a lot of copies has been made, what do you do next? You transform this organism, the host. You insert this DNA into a suitable host where this gene can get expressed. We know for gene expression to occur, the promoter must be inserted or the existing promoter should be used of an organism of the host, or an additional promoter is added at times, in most of the times though. So when you have the gene which is being placed inside the host cell, the organism that you end up getting is a transgenic organism, or you can say it's a genetically modified organism. This is produced. Now we have to identify whether the genetically modified organism has been produced or not. Because all these things, once they are being done, you have placed the gene inside the host cell, the host cell might not be transformed. The host cell might not have taken up the gene that you wanted it to be inserted into the cell. So we have to identify that. And when we have to identify this transformed cell, we use gene markers for the screening purpose. And the gene markers include GFP gene. This is green fluorescence protein. This gene allow this organism which is being transformed to glow green in this way. So you can screen and you can identify whether that has been transformed. So this organism which has been transformed will appear green, and those who have not been transformed, they will not flourish green. So once we have identified our genetically modified organisms, we take them out, we place them for in a medium where large scale production of the host cell can be done. And this host cell, keep in mind, is the genetically modified organism which is being produced by inserting the DNA. So how do you obtain the gene? Once the gene has been obtained, I discussed that yes, gel electrophoresis is carried out, but before gel electrophoresis, you have to take out the gene first from the cell. Now, how do you obtain the gene from the cell? You don't obtain it from gel electrophoresis. Yes, you identify it using gel electrophoresis. So how do you obtain the gene? The gene can be obtained either extracted from the cell, or the gene can be artificially synthesized using gene machines or DNA machines. So these are the two basic ways in which the desired gene can be obtained. Now, how do you extract the gene? When you're extracting the gene, you cannot take out the gene from the nucleus because the nucleus is having like two copies of that gene inside the nucleus, and the DNA is on the nucleus, the chromosomes. And that's not just one gene, the chromosome is having several genes present together, one on top of the other. So the gene locus is there. So it's not just one gene. For example, if this is the chromosome and you want to take out this particular gene from here, you have to cut this chromosome and take the gene out. There's a chance of cutting the chromosome at multiple positions. So what do? You take the messenger RNA from that cell because the messenger RNA will be in high quantity in the cell if it is expressing that particular gene. So a high quantity of that messenger RNA will be produced in that particular cell. From the cytoplasm, you can extract this messenger RNA. Once this messenger RNA is being like taken out, you produce a cADNA out of it using the reverse transcriptase enzyme. Now, how do we do this? We'll discuss this later on in the same video. So what do we do further? What is the first thing? The gene being extracted from the cell. The second thing could be you synthesize an artificial gene. How is the artificial gene synthesized? In this way, we know that we already know the amino acid sequence of the protein, the desired protein which is being made. So knowing the amino acid sequence, we can predict which type of codons have to be placed, which of the nucleotide sequence have to be placed one after the other. We can add those short fragments of nucleotide using DNA ligase and the DNA on the gene machine. We can add the nucleotides there, and according to the amino acid sequence, it will help you join all those nucleotides in a proper order. So it can be made in a way that now it will end up having the codon sequence for that particular amino acid sequence that you actually wanted for the desired protein. Now, let's discuss these, how the gene is extracted from the cytoplasm. When we're discussing extraction from, I've discussed this in my other video as well, how the gene is extracted from cytoplasm. So just review this once again. So when you're extracting the gene from the cytoplasm, how do you do this? This is extracted from the form of mRNA. You know mRNA is single-stranded molecule. So this is single-stranded mRNA molecule. This is the start codon and so on and so forth. What do? Inside the when you want to take this mRNA and turn this into the desired gene, what do? You add these ingredients together with the mRNA. You add DNA primer, you add free nucleotides, which are called dNTPs. You incubate them with the enzyme reverse transcriptase, and you keep adding further enzymes which are required according to the process. The first thing is you allow a DNA primer to attach to the free end of an mRNA. This DNA primer allows this particular part of the mRNA to become a double-stranded. When this becomes double-stranded, the double-stranded end will allow the attachment site for the enzyme reverse transcriptase to attach here. This reverse transcriptase enzyme attaches here and starts to move along in this direction. When it moves along the mRNA, it ends up producing a cDNA in this direction. What is cDNA? cDNA is the complementary DNA, a single-stranded molecule of DNA, which is a transcript of mRNA. You know the process of transcription is production of mRNA from the template DNA. But here you are carrying out reverse transcription. This is reverse transcriptase enzyme being used. In this, you will be producing a single-stranded cDNA using the template mRNA. But this enzyme cannot work if the mRNA was single-stranded alone. So you have to turn this end into a double strand using a DNA primer. Now, once this cDNA has been produced, you have the cDNA ready. But now one of your strand is mRNA and one of the strand is cDNA. What do? You have to digest this mRNA and remove it from here. So what do? You place an enzyme RNase. This degrades the denatures, or just not just denature cannot be used as a term here. It degrades and just destroys. This destruction occurs of this mRNA. So the mRNA gets removed. What are you left with? You are left with cDNA alone, which is a single-stranded molecule. Now you have to turn the cDNA into a double-stranded DNA molecule. So what do? Again, you attach a primer here. The primer is attached. It turns this end as a double-stranded molecule or double-stranded end. Now to this double-stranded end, the DNA polymerase enzyme will attach here, and the double-stranded polymerase, the double-stranded this DNA strand will allow the DNA polymerase to start moving in 5' to 3' direction, and elongation will occur, and you will end up having another DNA strand being produced against the strand that already was there. So by the end of this process, you end up having two strands of DNA. A double-stranded DNA is being produced. This double-stranded DNA is your gene. So you actually have produced the gene, you have extracted the gene using the codes from mRNA from the cytoplasm of a cell. Now, once the gene has been produced, this is the gene. Now you have to add this gene into a host cell. Now to add this gene into a host cell, you have to add overhangs on both ends of this gene. These overhangs are the sticky ends. You add free nucleotides of one sort on one end of the molecule, and on the opposite strand, on the opposite end, you add the same again. Now these sticky ends will go on and they will attach to the host cell where you will have the complementary sequence to these. So for example, if I have to place this DNA into a host cell, that host cell must have the sticky ends complementary to this end. So for example, the GGG, the nucleotide will be there, and then the structure of DNA will continue. So this DNA will get incorporated into the host genome because of the sticky ends. The sticky ends are being complementary on this side as well and on this side as well. So this is how this will get incorporated into the host cell DNA, and you will have the gene being placed inside a host cell. This is how a gene is added into a particular organism. You extract the gene, you add the overhangs or the sticky ends. Now, once we have the DNA ready, what other tools do we need? We basically need. The other requirements for we actually we have our restriction endonuclease, and this restriction endonuclease enzyme is used to cut the DNA segment. And when it is used to cut the DNA segment, it is this ends up cutting the DNA segment at a point called restriction site. And when it cuts at the restriction site must be a palindrome sequence. What's a palindrome sequence? A palindrome sequence is a sequence of bases which are same on both strands but in opposite directions. So for example, when you're going from 5' to 3' and the DNA reads TCG, TTAACGA in the forward direction, on the complementary strand coming from 5' to 3' in the opposite direction, you will have the same sequence of TCGTTAACGA, but in the opposite direction. So this is called the palindrome sequence. The palindrome sequence is the precise site where the restriction enzymes produce a cut. So for example, there is a particular restriction enzyme, it produces a straight cut. There can be another restriction enzyme that produces you can say, a jagged cut. Now, in these both ends, in these both cases, what's happening when a line of cut is straight, this ends up producing two ends which are blunt ends. Blunt end means there is no overhang being produced. It's just the phosphodiester bonds have been cut on both sides. But when the DNA is being cut along the staggered ends, or you can say the recognition site is a palindrome and sticky ends have to be produced. So the DNA will be cut in such a way that the phosphodiester bond will be cut, then the hydrogen bonds will be broken, and the phosphodiester bond will be broken again. So this ends up producing an overhang at this point, and the complementary overhang goes on the other side. So you are having the sticky end being produced on both ends. So when you cut the DNA can be cut either with a blunt end or with the sticky end. Now, if the blunt end have been produced, I've already discussed how the sticky end will end up being incorporated into the host cell. When you place this gene there, there will be a complementary sequence of the sticky end to which these will attach, and this will allow this DNA to be incorporated to the host cell. What about the blunt ends? The blunt ends cannot be placed without a sticky end being added. So once this is done, these blunt ends have to be converted into sticky ends by adding single strand of short stretch of bases on both ends. So these are called flank sequences. We don't discuss flank sequences in our A-level course, but anyhow, so you should know that blunt ends cannot be added to existing sequence of the gene without overhangs being added there, otherwise it will not attach. So moving on, this is the detail of gel electrophoresis. I've already covered this in detail in one of my videos, how gel electrophoresis is done. It's basically a technique to separate the molecule the base of size and charge. The DNA is separated and the protein can be separated. You place either one of those sample, either you will place DNA fragments or you will place protein and you separate them from the samples. For example, if you are using DNA, so you what will you do? You will just place the fragmented DNA into these gel wells. There are wells that you make, and on the wells you add the DNA sample. So if this is my gel, I'll have wells at one end, and in these wells I'll add my initially the control DNA, and in the remaining wells I'll add my DNA samples that I have to like identify which fragments have to be placed there or which size fragments are there in each of the samples. So these three, for example, are having my samples of mixed DNA fragments. I want to know which size of DNA fragment is there in each well. And the first well is having the DNA control or control ladder, or you can say the DNA ladder of known sizes or known fragments. Now, once this is settled, what I'll do, I'll place my just gel in a chamber filled with buffer. Now the control DNA ladder is added to the first, as I already discussed. The DNA fragment of different sources have to be added in separate wells. You add the DNA using micropipettes. We add glycerin and fluorescent dye. The glycerin is added so the DNA sync to the well. Because if you look at the gel, for example, if this is my gel and this is the side view of the well, so the DNA, if I allow the DNA will be here somewhere at the top. So the DNA can come out of the well when I place it in the buffer solution. So I must allow this DNA to go and sink to the bottom. And how do this happen? The glycerin being added. So once the glycerin is added, is allow the DNA to settle at the bottom of the well. So once the DNA has been settled, for example, this is my well again, the DNA has been settled in this position here. So when I apply a potential difference, the DNA will start to travel in a straight line. Depending on the size, if it's a smaller size DNA fragment, it will travel to a longer distance. If it is a larger size DNA fragment, it will travel to a shorter distance. So the fluorescent dye will eventually help me identify the DNA segment, the DNA fragment to the point where it has traveled. What would be the magnitude of the electric current? I can apply 50 volt for 30 minutes. Once this has been applied, my gel will have the DNA separated. So if I look at my gel, I'll take it out of the chamber. I'll place it in the UV chamber. And these were the four wells, for example. I'll have a DNA ladder of this sort because it will be visible to me since I added the fluorescent dye there. Now my DNA fragments of different sizes will be seen here as well. So every fragment basically tells me that, for example, this particular well had a had actually a fragment of DNA that had a size equal to the one which was present in the DNA well. So this is how we identify the DNA fragments. And once the DNA fragment has been identified, I can place this DNA fragment into a PCR machine and I can amplify this and make a large quantity of this before placing it inside the host cell. So this is how this is done. This is something that already I have covered in detail, how the gel is made, how you take this particular this compound, either agarose or polyacrylamide, you add it in warm water to make a solution. And once this has been done, these are the wells in which you place your DNA sample after placing it in the buffer. The cathode is always attached near the well, so it allows a negative charge to be here near the DNA, which itself, the UV node, the DNA is also negatively charged. So the negative charges, they repel each other and the DNA starts to move towards the positive terminal of this particular region towards the anode. So the DNA fragment, which are of larger sizes, they get separated from the ones which are smaller ones. So for example, if this DNA ladder was here, my large size DNA fragment of control, for example, 200 kilobases DNA would stay here, whereas 10 kilobase DNA would travel a longer distance because it was a smaller fragment. So for example, I want to identify the third sample that I had. What is the size of this particular DNA fragment? What I'll do, I'll just compare the distance to which it has traveled according to my DNA ladder. So if it has traveled the distance same to the one which was 50 kilobases, so this DNA which is here is 50 kilobases DNA. So this is how we can separate the DNA and we can compare them with one another, how this goes. And this can be like picked from here and then placed in the PCR as I've already discussed. What do you do in the PCR? This is the technique used to amplify the DNA. We use a polymerase chain reaction. How the polymerase chain reaction is done, this is due to amplify the DNA. We have a thermocycler. This thing is the thermocycler here. This is the fifth thing. There are five things that you will need here for a PCR to run successfully. I have a thermocycler machine with me. This is the thermocycler machine. I'll place 1, 2, 3, and 4 inside a tube inside this thermocycler. Now, what's 1, what's 2, what's 3, what's 4? Let's discuss this. So this is the thermocycler. Inside the thermocycler, what is a thermocycler? Thermocycler is a computer-operated machine that varies the temperature over a period of time. So I have this machine with me. I'll take a tube from this machine, and inside the tube, inside this, I'll start to place my DNA fragment that I want to be copied. The one that I did just took out from isolated the cell, and then I got it from, for example, this was the DNA fragment, the size that I wanted. I'll place it, I'll take it out and I'll place it in PCR machine. So this is the DNA fragment to be copied. Together with this, I'll add the DNA polymerase, which is Taq polymerase. Now, a Taq polymerase is a thermostable polymerase. This is taken from an archaean. What is an archaean? Archaean is an extremophile prokaryote. The name of this prokaryote is Thermus aquaticus. You already have discussed the binomial nomenclature. You must be aware that the name of the organisms in scientific terms are given by the binomial nomenclature. So the name Thermus aquaticus shows that this particular organism, the species Aquaticus, and its genus is Thermus. So from Thermus aquaticus, we take this Taq polymerase. This organism lives in hot water, in hot springs, so its enzymes do not denature at high temperature. So we can keep on adding nucleotides, it keep on and heating it so that the DNA keeps on getting copied again and again. And this enzyme will not denature because we actually have to place it in a thermocycler. We cannot place the human DNA or the mammalian DNA polymerase there. Sorry, not DNA, the DNA polymerase. The DNA can still, the DNA can withstand the high temperature, but the DNA polymerase cannot. So we have to place a specialized DNA polymerase called Taq polymerase, which is thermostable. So after that, we have to add primers. We already have discussed what primers are. These are short stretch of bases that attach to the 3' end of the DNA. They produce a double-stranded end, so the DNA polymerase can then attach and bind to the DNA and then elongate the DNA. Then what's the fourth thing we have to add? We have to add DNTPs, free nucleotides. They have to be added to of all the four bases, A, T, G, C. We have to add them in excess. Now, once all these things are in place, I have them in my thermocycler. I set my thermocycler for the number of cycles that I want. For example, I'll just start 100 cycles. So one cycle will have 3 steps. So these 3 steps will go 100 times. And when they keep on occurring 100 times, so this will amplify that particular DNA fragment exponentially. How will that occur? One cycle will have 3 steps. A single cycle is having 3 steps. The first step is denaturation. Denaturation occurs when you heat the DNA. We know that the DNA we have is a double-stranded DNA. We have to convert it to single strand by heating it. With that breaks the hydrogen bond between complementary bases. So you heat it to 95 degrees Celsius to break the DNA from the double strand and convert it to a single strand. Then second step is annealing. At annealing, you lower the temperature to 65 degrees Celsius. When you lower the temperature, this allows the primer to attach to the 3' end of the single-stranded DNA. Once the primer is attached, the next thing is you allow the DNA molecule to elongate using the DNA polymerase, Taq polymerase. We know that now the DNA Taq polymerase can elongate because the primer is already attached. So this gene will start to get copied. So this one cycle will end up producing 2 copies of the same DNA molecule. So the next cycle will start with 2 DNA molecules. So this is how diagrammatically I can represent this. As for example, if this was my DNA sample that I wanted to be copied, it's a double-stranded DNA. So this is 3' and a 5' end. The opposite, the other strand will be anti-parallel 3' at the bottom and 5' at the top. So I have done a process A here. So this process A include 2 different steps here at the same time. I have separated the DNA fragment, the double-stranded from one another, which means I've carried out denaturation. And at the 3' end, I have attached the primer as well. So this is annealing occurred together with the denaturation. Now, after this, I start to add my DNA Taq the Taq polymerase, the DNA polymerase. It attaches to the double-stranded end and it starts to elongate the DNA along 5' to 3' direction. And towards the end of one cycle, I'll have the DNA produced in twice the quantity as the first. Now, since it's a thermocycler, it will repeat the same 3 steps again and again, but this time it will start from the 2 DNA molecules. So it will convert these 2 DNA molecules and double them further. So every time the cycles keep on going on, we have the DNA being produced in exponentially large quantity. So that was the aim of PCR. We amplify the DNA fragments using PCR. So once we have this thing ready for us, we have the gene amplified. We will place this gene and place it with the sticky ends in the DNA. For example, if I'm using the plasmid as a vector, so what I'll do, I'll use the enzyme to cut this DNA fragment. So this is, for example, the plasmid that I want my gene to be inserted in. So what I'll do, I'll take the bacterial plasmid. This bacterial plasmid is there, and I'll cut the DNA, the plasmid. We know the plasmids are circular DNAs. So when you have a ring of DNA, which is a plasmid, we know that you can cut the DNA open and you can place the desired gene inside it. How so? So for example, if I have this particular plasmid with me and I have to, this is a ring of DNA, or you can say the circular DNA. So I have to first produce a cut here. So I have to use a restriction enzyme to produce a cut. When the restriction enzyme produces a cut here, it opens this DNA at a particular point, the restriction site. And this is the restriction site at which I'll then place my gene that I took out from the process of, for example, I've just discussed, either the gene was extracted from the cytoplasm from an mRNA or was made using a machine, or then it was isolated using the gelileurophoresis and amplified with PCR. You place it with the plasmid. Also, you add the DNA enzyme ligase. The ligase enzyme allow this DNA to be placed, and when the sticky ends they join together, this gene gets incorporated. Now, together with the desired gene, we have to place a screening gene like the GFP gene marker. So this bacteria, when it takes up this plasmid bag, it will start to fluoresce green. We can screen it that yes, okay, this is my transformed bacteria. Apart from the GFP gene, we will have the promoter added as well. The promoter will allow the RNA polymerase to attach so the gene transcription can go on. What make plasmids suitable for genetic technology? Plasmids are small in size, they lack histone, they replicate independently of their host cell, they can be easily taken up by the bacteria cells. The DNA sequence can be recognized easily because there are fewer genes. There are various recognition sites for multiple restriction enzymes. Although we use just one restriction enzyme at a time, we don't want our plasmid to be cut at various restriction sites. Otherwise, we'll fragment the restriction, this particular plasmid altogether. Once this has been done, we know the gene transfer has occurred. We'll allow the gene to be expressed. The recombinant DNA has been placed inside the bacteria. The green fluorescent protein will be added. This will allow this bacteria to fluoresce green. The plasmid is inserted back into the bacteria using a process called electroporation, in which you place this bacteria in a calcium chloride solution. You give it heat cold treatment that open up small pores so this particular plasmid can go inside. Once this is that the bacteria has been transformed, you allow this to be placed in UV chamber so you can see which ones are fluorescent green. For example, there are so many bacteria in this particular view field of the microscope, and then there are these few which are fluorescent green. These are the ones that have taken up your gene. So we take out those and we place them in fermenter and we grow them. Apart from plasmids, we can also use yeasts instead of bacteria as a particular source of vector. Why can yeast be preferred over a bacteria? Because yeast is a eukaryote. It will modify the protein very similar to the ones which are modified in the human cell. Because yeast have Golgi, yeast is having ATS ribosome, it is having transcription factors very similar to the ones which we have in the animal cell. So we have discussed these enzyme, reverse transcriptase, restriction endonuclease, RNase, DNA polymerase, DNA ligase. What are restriction endonuclease? These are the enzymes that cut the DNA at a restriction site called the palindrome site or the palindrome sequence. And we have discussed all these enzymes. So this is this example how you can take this plasmid out of a bacteria, cut it open, you have the human insulin gene, place it here together with the GFP, the gene is added together with the promoter, and then you can add it back into the bacteria. So this is how this process is done. I hope this video of covering the principle of genetic technology was helpful for you in understanding the basic principles which are applied when we are discussing genetic technology. Thank you. I hope that will help you getting these concepts through.